Okay. Today's topic is urea cycle. This is one of the metabolism in amino acids. Is my voice audible to you people? Yes?
Yes, ma'am.
Okay.
Yes, ma'am.
So the topic here is urea cycle. So this urea cycle will be dealt in amino acid metabolism topic. So the contents which include are introduction to urea cycle. What are the different steps that are involved in urea cycle? What is the energetics and how can we regulate this urea cycle? And what can be the different metabolic disorders if this urea is not maintained properly in the body? That means it is not excreted properly. So let us see the introduction to urea. So urea, as you all know, it is nothing but it is the end product of the protein metabolism. So this protein metabolism occurs in humans as well as mammals. So in protein metabolism, you have nitrogen which is present in the amino acids, that is NH2 group present in amino acids, amine group. So amine groups is containing nitrogen, so that nitrogen will be converted into ammonia in the body, but that ammonia concentration is increasing in the body, means it is leading to some toxicity to the body, so it has to be eliminated. So it will be eliminated through fecal matter, and it accounts for about 80 to 90% of the nitrogen containing substances. That is, urea is accounting for 80 to 90% of nitrogen containing substances, and it is mainly seen in the form of ammonia. So that ammonia is mainly obtained by the product of oxidative deamination reaction, and here even small quantities which are present in the body, they also have to be excreted from the body. So the urea whichever is there, it is transported to the kidney and it is excreted. So this is the structure of urea: carbon with keto group, and it has two amine groups. So that is the structure of urea. So for the production of urea, there should be a starting material and the ending material. The starting material is nothing but ammonia and carbon dioxide. So on metabolic processes, it will be converted into urea. So the overall urea formation reaction can be given like this. That is, we require two molecules of ammonia, carbon dioxide, because these two are the starting materials. And for the metabolic processes, you require, for the reaction to exist, you require some kind of energy, which is nothing but ATP. So it requires three molecules of ATP, and hence it is getting converted into urea, which is nothing but the end product. Along with urea, there is a liberation of water molecule and some amount of energy which is in the form of ADP. So this is the overall urea formation reaction. That is nothing but ammonia and carbon dioxide are the starting materials. These two are combined in the presence of ATP molecule. They are giving urea and water along with ADP. So this is the introduction part of urea. We will see how this urea cycle is taking place. So this urea cycle is also known as Krebs-Hanslett cycle. So this cycle is simply can be called as Krebs-Hanslett cycle, where there will be a conversion of toxic ammonia into non-toxic urea. That means the starting product, I was telling that it was ammonia and carbon dioxide. So it requires that ATP energy to convert into ADP and urea formation along with water liberation. So here what will happen means there will be a conversion of the toxic material, which is nothing but ammonia, which is obtained by the oxidative deamination, and that will be converted into non-toxic urea. And this majority of the reactions will be taking place in the mitochondria, and some part of the reactions will take place in the cytosol of the cell. So here what will happen, you have seen the structure of, here is the structure of urea: carbon attached with the keto group and double bond O, and two amine groups are present. So these two amino groups are obtained from each of the amino acid, which is nothing but one amino group is coming from the deamination of glutamic acid. That is, in glutamic acid, nitrogen group is removed by by oxidation process, which is known as oxidative deamination, and that amino group is attached here, and other amino group is coming from the aspartic acid. So this aspartic acid is generally obtained from the fumaric acid, which is produced by the urea cycle. So whatever the fumaric acid which is produced by the urea cycle, it is undergoing series of reactions, and it is also entering into the Krebs cycle or the TCA cycle. And in TCA cycle, you can see the formation of oxaloacetate. So here there will be a formation of oxaloacetic acid, which is converting into aspartic acid. So what happens here in urea cycle means the ammonia whichever is there, which is in toxic form, it is converted into non-toxic form, and the major portion will be taking place in the mitochondria and some part will be taking in the cytosol. And since the structure is containing two amino groups, one amino group is coming from the oxidative deamination of glutamic acid, and the other amino group is from the aspartic acid. So what happens to this aspartic acid means this aspartic acid is mainly produced from the fumaric acid, which is given in the urea cycle. So this fumaric acid will then enter into Krebs cycle, which is nothing but citric acid cycle, and it will form oxaloacetic acid. This oxaloacetic acid on the reaction with the transaminases, and it undergoes a process known as transamination. It will combine into aspartic acid. So this is the introduction of urea cycle. We shall see what are the different steps that are involved in urea cycle. The first one, these are the 5 steps. The reaction or the steps which are involved in urea cycle are 5. The first one is synthesis of carbamyl phosphate. That means you are going to synthesize carbamyl phosphate, and this carbamyl phosphate will be converted into citrulline, and then from citrulline it will be converted into arginosuccinate. Once the arginosuccinate it is formed, it will be cleaved, and on cleavage it will lead to the formation of urea. So these are the 5 steps which are very much essential. Let us see the each step in detail. Step 1. So in step 1 is nothing but the synthesis of carbamyl phosphate. This carbamyl phosphate synthase, which is known as CPS1. So CPS1, it is an enzyme which is present in the mitochondria. It will catalyze the condensation of NH4 plus ions with carbon dioxide to form carbamyl phosphate. That means here what will happen during the condensation of ammonium ions with carbon dioxide in the presence of CPS1 enzyme, it forms carbamyl phosphate. So here what will happen, it will require some amount of energy. That energy will be given in the form of ATP. So in this step it will require 2 ATP molecules, and it is a quite irreversible and rate limiting step, which is very much essential. And this CPS1 which is there, it requires an acetyl glutamate for its activity. So CPS1 is there and CPS2 also is there. CPS1 is very much essential because it combines with the it is useful for the catalysis of the ammonium ions along with the carbon dioxide, and it also requires 2 ATP molecules. So another enzyme is there which is known as carbamyl phosphate synthase 2, CPS2. So this is mainly involved in the pyridine synthesis, and it occurs in the cytosol. So this CPS2 whichever is there, it will accept one amino group from the glutamine, and it does not require N-acetyl glutamate, which can be simply given as NAG. That means this is N acetyl A and G, NAG for its activity. CPS1 requires NAG, whereas CPS2 does not require NAG. So now the synthesis of carbamyl phosphate is done. How is the synthesis done? Because in the presence of an enzyme and also uh, ammonium ions on treatment with carbon dioxide. So the second step is formation of citrulline. So whatever the CPS which is produced, that has to be converted into citrulline. So how is it converted means whatever the citrulline is there, that is synthesized from CPS, which is nothing but carbamyl phosphate and ornithine by ornithine carbomylase. So using this enzyme, it is got synthesized. So here the activity of ornithine is very much essential and it is regenerated, which is further used in the urea cycle. So here the activity of citrulline is very much essential and it is almost similar to that of the oxaloacetic acid, which is important in the citric acid cycle. So ornithine and citrulline, these are the basic amino acids which are never found in the protein structure due to lack of codons. That means these ornithine and citrulline, they are very much essential amino acids which are basic in nature, and their structure is not much found because it is having the lack of codons. And this citrulline whatever is produced, it is now transported to the cytosol by a transporter system. Using some transporter system, it is sent from mitochondrial space to the cytosol. So the step 3 is synthesis of arginosuccinate. So for now citrulline has been produced with the usage of ornithine and ornithine carbamylase enzyme. So now arginosuccinate has to be synthesized. So the third step is the synthesis of arginosuccinate. How is this arginosuccinate synthesized? Whatever the arginosuccinate which is going to produce, it requires an enzyme known as arginosuccinate synthase. Synthase means which is useful for synthesis. So here what happens in the second step, whatever the citrulline which is produced, that citrulline along with aspartate, it will convert into arginosuccinate in the presence of an enzyme arginosuccinate synthase. So the third step is nothing but the synthesis of arginosuccinate, which is mainly done by the reaction with the citrulline and aspartate. Citrulline is found in the second step of the urea cycle. So what happens till now, we were telling only one amino group is got incorporated. Now the second amino group is also incorporated in the reaction. So this reaction is also very much essential. The third step is essential because here ATP whichever is there, it is got cleaved into adenosine monophosphate and pyrophosphate. Here what happens means whatever that ATP energy is there, that is got cleaved into AMP and pyrophosphate, and later it is broken down into inorganic phosphate. So the first step, 2 ATP molecules is required, and here ATP is got converted into AMP and pyrophosphate, which is again equal to 2 ATP molecules. So the cleavage of arginosuccinate. Now how did this arginosuccinate be produced? You have citrulline which is given in the second step, that is combined with aspartate in the presence of synthase enzyme. You got arginosuccinate. Now that arginosuccinate is to be cleaved. So what will happen? Whatever the arginosuccinate is there, in the presence of arginosuccinase, it is a kind of enzyme. Using that enzyme, it is converted into arginine and fumarate. That means whatever the arginosuccinate which is produced in the third step, it is converted into arginine and fumarate, and this arginine is the immediate precursor for the urea. That means for urea synthesis, this is the immediate precursor. So the reaction will take place in the presence of arginosuccinase enzyme. Now, I was telling you that arginine succinate is produced in the third step, and now in this fourth step, it is got cleaved. So, on cleavage, it is giving, what is it giving? It is giving arginine and fumarate. So, this arginine is important for urea formation. What will happen to this fumarate? It will enter into the TCA cycle or the citric acid cycle. Thereby, it is providing a connective link with the TCA cycle and gluconeogenesis. So gluconeogenesis is nothing but production of glucose from non-carbohydrate sources. Now we have arginine, which is obtained by the cleavage of arginosuccinate. Now the fifth step is the formation of urea. Let us see how the urea is got formed. So whatever the arginine which is there in the presence of an enzyme known as arginase, this arginine, in the presence of arginase enzyme, it will be converted into urea and ornithine. So this ornithine is regenerated and then it can be reused in the urea cycle. So this arginase, which is the enzyme which is got incorporated there for the production of urea, it will be activated by some cofactors. That cofactors are nothing but CO2 plus and Mn2 plus. And ornithine and lysine, they will be competing. So here, arginine is very much essential for the formation of urea. Ornithine and lysine are also there, which are competing with arginine and they are showing competitive inhibition. So this enzyme, whatever the arginase is there, it is activating by some cofactors known as CO2 plus and Mn2 plus, and they are actively competing with certain amino acids such as ornithine and lysine. And this enzyme, which is nothing but arginase, it is mainly found in the liver, where some part will be entering into the urea cycle and some will enter into the tissues. So for this region, what will happen? Whatever the arginine synthesis is taking place, it may occur at it may occur in various degrees in many tissues. That means arginine synthesis can occur in many tissues, but for the production of urea, only the liver can ultimately produce urea. So this is the steps. The first step in the urea cycle is nothing but synthesis of carbamyl phosphate. In the presence of carbamyl phosphate synthase, it is an enzyme, CPS1. And the second step, what will happen means, whatever the CPS, which is carbamyl phosphate, which is produced, that should be converted into citrulline. That means there is a formation of citrulline in the second step. Third step, this citrulline along with ornithine, it will combine to give arginosuccinate. So that arginosuccinate will get cleaved in the fourth step. So ultimately, when it is cleaving, it will go ornithine and it will be giving arginine and fumarate. This arginine is very much essential for urea synthesis, whereas this fumarate, whatever is produced, it will enter into the TCA cycle. So let us see the energetics. So in energetics, we will talk about that it is a irreversible process. That means no backward flow. Backward it cannot go, and the total ATP which are consumed are 4 ATP. That means 2 ATPs are mainly required for the synthesis of carbamyl phosphate. That is, in the first step you require 2 ATP, and in the third step, that is for the conversion of arginosuccinate, to produce arginosuccinate. In the third step, synthesis of arginosuccinate, here ATP will be converted into AMP and pyrophosphate. So total 4 ATP. So let us see how the urea cycle is. This is how the urea cycle is. You can see here carbon dioxide and ammonia in the presence of carbamyl phosphate synthase, which is nothing but CPS1. It requires N-acetyl glutamate, NAG, for its activity. So here ATP is converted into ADP. So the first step is synthesis of carbamyl phosphate. So this is the first step. And second step is formation of citrulline. So this carbamyl phosphate will be converted into citrulline, This 1 and 2 reactions will be taking place in the mitochondria, whereas the third step is the formation of arginosuccinate. So what happens here means arginosuccinate synthase. It is a kind of enzyme where ATP is converted into AMP and pyrophosphate. So here arginosuccinate is produced. This arginosuccinate, in the presence of arginosuccinase enzyme, it is cleaved into arginine and ornithine, arginine and fumarate. The fumarate will enter into the Krebs cycle. Arginine, whichever is there, it is mainly essential for the synthesis of urea. So this is how the urea cycle is taking place. So 3, 4 and 5 reactions, they occur in the cytosol of the cell. So let us see how the urea cycle is regulated. So mainly the urea cycle will be regulated by the first step, which is nothing but catalyzation or synthesis of the CPS1. Synthesis of CPS1 is taking place. Synthesis of carbamyl phosphate is taking place, which is nothing but by it is done by the enzyme which is known as CPS1. So the first step catalyzed by the CPS1 is the rate limiting step in urea synthesis. And this CPS which is there, which is nothing but carbamyl phosphate, it is activated by N-acetyl glutamate, that is nothing but NAC. That means for its activity it requires NAG. So here what will happen? The high concentration of arginine, it causes increased levels of NAG, leading to the enhancement of urea synthesis. So NAG activity is to act on CPS. CPS1 will increase means arginine level also will increase. Then urea synthesis can be enhanced. So let us see how is this NAG synthesized. The NAG, N-acetyl glutamate, is synthesized from acetyl-CoA and glutamate. That means by the combination of acetyl-CoA and glutamate, NAG is produced, and it is done and it can be degraded by certain enzymes which are known as hydrolases. So the very much activity will be shown by CPS1. CPS1 and glutamate dehydrogenase, they are mainly applicable in the mitochondria. So when can we say that person's energy levels or ammonia levels are increased? So when a person is taking high protein diet, it means that the ammonia level is increasing because proteins on degradation, they give ammonia, which gets converted into urea by a urea cycle. So it simply says that a consumption of high protein diet, it can increase the level of NAG. So this is about the regulation of urea cycle. Let us see what were the different metabolic disorders. Metabolic disorders are nothing but if the metabolic processes are not taking place in the body properly, there are some diseases or defects which can occur. So here we are listing some 4 different disorders or defects, and what is the enzyme which is involved, and what can be the different characteristics which they show. The first one is hyperammonemia. Ammonemia means increase in the concentration of ammonia. So that can be exhibited by two types. One is type 1 and type 2. Hyperammonemia type 1. So this hyperammonemia type 1 is mainly because of the enzyme known as carbamyl phosphate synthase 1. So if this enzyme is involved in that reaction, this can occur. And what are the characteristics? In blood, high ammonia rate will be seen, because of which mental retardation of the children is observed. And hyperammonemia type 2. So hyperammonemia type 2, it is a kind of ammonemia where there will be an increased levels of ammonia in the body. So what will happen means it is mainly done by the enzyme ornithine transcarbamylase. This is the enzyme which is involved, because of which what will happen means High ammonia and glutamate levels are generally seen in the blood as well as the biological fluid, which is urine. So first and second are the types of hyperammonemia, type 1 and type 2. The second is, the next is citrullinemia. Citrullinemia is nothing but a condition where the synthesis of citrulline is high in the body. So in urea cycle, we were talking that citrulline formation is the second step. That means for the for whatever that carbamyl phosphate which is produced in the first step, it is getting transferred into citrulline. So if this citrulline concentration is increasing, we call the disorder as citrullinemia, which is mainly because of arginosuccinate synthase. And high ammonia and citrulline conditions are seen in the blood and urine. The next one is hyperarginemia. So the name itself is telling there is an increase in the concentration of arginine. So it can be done by an enzyme known as arginase. And the characteristic features are there will be a high arginine concentration in the blood. CSF is nothing but cerebrospinal fluid and urea. So these are the different metabolic disorders which are occurring because of the urea concentration in the body. So hope you understood.